This is the trypsin activity in response to environmental changes in pH project. It was done for the class of Bio 200 Lab, Section 6, Towson University. This is my abstract. Introduction. So, to start this off, enzymes are basically proteins, catalyst proteins, that help increase the rate of chemical reactions. So they basically help speed up chemical reactions. And the special thing about them really is that they do not get altered during that process. They stay the same throughout the whole process. So the way that enzymes speed up chemical reactions is basically they lower their activation energy of the chemical reaction, and they bind to the reactants while that's happening. So enzymes are crucial to living organisms, not only because they help speed up chemical reactions, but they also regulate to help cells. So on the long run, cells respond well in different conditions they're put on. So, pH changes, temperature changes. So enzymes really help regulate that stuff. So trypsin, of course, what we're talking about in our introduction, as you can see, trypsin is a protease. It's found in our pancreas and small intestine, as stated in the board, of course. We hypothesize that the optimal environmental conditions for maximum trypsin activity would be a pH of 9 and an incubation of 57 degrees Celsius to show how trypsin was actually affected by pH changes. So to do that, we tested pHs, not only pH 8, 9, and 10. We actually went in half, so 8.5, 9, 9.5, and 10. So we would actually see if these half increments would actually help us get the optimal enzyme activity at, of course, 57 degrees Celsius. And our question was that how does pH level affect enzyme activity? So this is really what we were looking for. So for our methods, the reaction buffer that we used was NaCl and tris hydrochloride on the pH, which we ranged from 8, 8.5, 9, 9.5, and 10. Since we added 450 microliters of the reaction buffer we needed, the half pHs that we did was basically we added 250 of each buffer to, let's say, it's 8. So what we did was half 450 microliters were needed. So we used 225 microliters of pH 8 and 250 microliters of pH 9, equaling 450 microliters to create 8.5. So we put 500 microliters of the substrate, which was azocasein. The substrate was actually designed by chemically adding sulfanilamide groups to the milk protein casein. And these sulfanilamide groups, which are orange, are attached randomly to the casein. So after that, 50 microliters of the enzyme trypsin, we added that to each pH to basically break down the azocasein. So we of course had negative control groups because we always want to have a control group to make sure that we are actually seeing the difference. So for our negative control, instead of adding trypsin to the mix, we actually added water. We added 50 microliters of distilled water into that mixture. So basically to do that, just so we can have an equal experiment from both sides, and an amount for both sides. So we kept the volume in the tube and put them all in the centrifuge in the centrifuge machine to make sure that we will break down the substrate. So we did that because trypsin was the cause of the breakdown of the substrate. So in our experiment, we designed 2 replicates of negative control and 3 replicants of actual active controls, which are basically the trypsin ones. We use these replicates because there is a variability between experimental units. So basically, what happens is when you only have one negative control group, sometimes you might do something wrong and that will make you think that the negative control is supposed to be like that, but it really isn't. So this is why we use like multiple control groups, just so we can compare the average and kind of get the correct or the closest answer. So every single tube was incubated after that at 57 degrees Celsius for 20 minutes. We actually use different timers for each group. So we basically put in trypsin after we put in the azocasein, and then we incubated for 20 minutes the first group. Once that group is finished, then we incubated the second group that we were working on while that group was incubating. So we constantly kept the tubes in the correct amount of like wait time and stuff like that. So we actually really paid close attention to that. We did write down everything in our notebook. So we had like a specified chart in our lab notebook that had everything that we got from the 20 minutes of incubation. So right after incubation, we added 500 microliters of TCA. So the reason we use TCA is because it helps stop the reaction that, trypsin is breaking down azocasein. So TCA stops trypsin from breaking down azocasein. So that is the role of TCA. So each tube then was centrifuged at 14,000 RPMs for 2 minutes. The reason we do that is because we want to separate the undigested azocasein. That will basically be like a little pellet at the bottom of each centrifuge tube. And then we will have the supernatant, which is what we measure for absorbance, which I will speak about after this. So after we did that, the digested azocasein was bound to the liquid supernatant. So the way we wanted to know the absorbance, which is what we did next, was by putting it in the spectrophotometer at 440 nanometers. So the spectrophotometer basically measures absorbance. We use a supernatant that we have in our centrifuge tubes and apply it in a glass, and that glass can actually, like, we place it inside of the spectrophotometer, and it will tell us the absorbance of the supernatant. So the sulfanilamide, the colored group bound to the substrate, allows azocasein to be absorbed at 440 nanometers. So the higher the absorbance we have, the darker the azocasein was digested. So basically it means that more enzyme activity was actually there. So for our results, trypsin was actually most active at 57 degrees Celsius at pH 8. The activity was like the highest, and the second highest was 8.5, and the p-value between these two pHs was about like 0.02, which is actually very small, and that is great news for us. Increasing the pH from 8 to 8.5 actually decreased the trypsin activity by about 35%, which was a surprise. But like the total enzyme activity fell about 70% after increasing pH by 2. So it's really a large number, but the activity did not stop. So it basically gradually decreased, but it did not stop. The activity of trypsin was actually slowly diminishing down with about 25% every time we go from either side of the pH spectrum, but it did not stop. The standard deviation was actually very large at pH 8. It was about 13%, and the other pHs were like 2% difference. The reason that happened was because we did like a human error, which I will speak about in our discussion. To summarize the human error that we did, we basically spilled a centrifuge tube, but we placed it in the next group for incubation. So like we redid it and put it in the next group, but we kind of feel like that was the reason. Yet we don't know if that really was. But it did not affect like the other pHs. So that 13% standard deviation was actually caused by human error from what we believe. So these are our results. As I spoke about the standard deviation right here, it was pretty large, and then the other were like about 2%. This is the numbers, as you guys can see, 0.13, and then all of them were like 0.0. 0 or like 1. These are our p-values. So we use the spectrophotometer to measure the enzyme activity at 440 nanometers. We use t-tests to show the true significance of trypsin activity in consideration of environmental changes in pH. Activity at 57 degrees was very different between all pH levels, like 8 to 10, because the p-values were all less than 0.03. The thing about that is if the p-values were 0.05 or higher, then it wouldn't be correct. So all of our p-values were actually very well. So in conclusion, trypsin did not get affected by the pH change. So this basically speaks about what I just said. We used the spectrophotometer. We found that at 57 degrees, the pH levels were actually very different. Like, the p-values were significantly different. Not significantly different, I'm so sorry. So for our discussion, which is the last part of this presentation, the experimental design and execution were adequate to test our hypothesis because our negative controls proved that it was in fact the trypsin breaking down the substrate. And the only variable that was being altered was the pH level, and that's what brought changes in our enzyme activity. So our hypothesis of a pH of 9 being the optimum was rejected because it was, we actually hypothesized it would be pH 8. So it wasn't really wrong. We just believed that it was going to be the highest at 9, but it resulted to be 8. The t-test proved significant changes because our p-values were less than 0.05. Enzyme activity was diminishing slowly by about 25% as we keep going on each side. So let me just speak about, so we were comparing results of other groups from the last experiment, and they actually thought that, well, concluded that at 57 degrees Celsius, they had the optimal enzyme activity. Their independent variable was the temperature. So we actually decided to change our temperature to 57 degrees Celsius, and we tested our pHs from last time, but we added the half increments, of course. So this is why we believe like our control variable conditions were like, you know, we thought that they were true because, you know, other groups had almost the same results. So we did have sources of variability. We had some human error. We did not add enough TCA as we believed to stop the reaction, and we also believed that when we dropped the centrifuge tube and had to redo it, we actually should have like, it could have like caused some variability, but you know, it remains to not be such a big thing because our other like results were not bad. They were actually really well. So that we do not know about for sure. If we were to do it again, we would definitely make sure we're extra careful, of course. We would maybe centrifuge our tubes one more time because we did have some pellet in our supernatant. We would also we would use the half increments again to just see how like the slight changes were affected, you know, throughout. So like basically instead of 8, 8.5, 9, 9.5, 10, we could do like 7.5, we could do 10.5 and kind of see where that would play. So for future experiments, we would test a few half incremented pHs below 8 to see which one is better. And lastly, our conclusion from this experiment. So we actually thought it was relevant to the ideal conditions to grow bacteria because bacteria is the best in like neutral pHs and warm temperatures. So it's more in the basic side a little bit. Our results prove this by showing that like our trypsin activity performs well at pH 8 degree at 57 degrees Celsius. So the trypsin we were using in this lab was likely used from a bacterial organism. So these are our references. Thank you.